Hi, welcome to biology classes with Lakshmi Chandra. In the previous videos, we were learning about how we can introduce our gene of interest without a vector. In today's session, I'll tell you how we can make multiple copies of our gene of interest without a vector. Okay, so there is no need of cloning vectors for cloning. Without a cloning vector, how we can do this process of cloning? Or in other words, I can use amplification of our gene of interest. Amplification means getting multiple copies, like we take Xerox copies, no? Same way. So amplification of gene of interest in vitro, that's our topic for today. The simple method used for that is polymerase chain reaction. Polymerase chain reaction is the simplest method that is done for amplifying the gene of interest in vitro. This is a very simple method. In this session, I'll tell you regarding the requirements for this method, and the next session, I'll explain to you the process. So let's just see the requirements. We need primers, dNTPs, Taq DNA polymerase, DNA template, and a PCR machine, polymerase chain reaction machine. Now, what is this PCR term is very familiar. We do this RT-PCR test and all for this disease corona to identify. Okay, so as students of biology, we should know regarding what this PCR is. Okay. Basically, dear students, please understand this, that this PCR method is based on the method of DNA replication, because we are getting multiple copies of our gene of interest. So the basic process is like our DNA is getting multiplied or DNA is getting replicated. So if you are not familiar with the DNA replication process, please refer to that session which I have already put regarding DNA structure and DNA replication methods. Okay, please go through that so that you will understand the base, and then that will help you to understand this particular PCR very clearly. Okay, coming back to our topic, today I am just taking you requirements of PCR. First one is written as primers. They are primarily needed ones. Okay, then dNTPs, Taq DNA polymerase, DNA template. So let me first draw here a DNA template. This DNA template, as we draw, we have to write the direction 5' to 3', and this is another. So this two together, this is a DNA double helical structure. I have just drawn it just to represent. I have just represented it here like this: 5' to 3', 3' to 5'. This is a DNA template. Okay, you know, when replication takes place, what the enzyme that is required for DNA replication is DNA polymerase. But here for PCR, instead of normal DNA polymerase, we use a DNA polymerase called as Taq DNA polymerase. Taq, T-A-Q. This stands for Thermus acquaticus. This is name of a bacterium that is present in conditions which are very high, inhospitable because of high temperature. So these are bacterium which they habitate areas having very, very high temperature. They are found in regions like hot springs, volcanic eruptions and all, where the temperature is very high. So their DNA polymerase can withstand this high temperature. Okay, so we have taken the DNA polymerase from this organism Thermus aquaticus. So the DNA polymerase is called as Taq DNA polymerase, and the speciality is that this DNA polymerase can withstand very high temperature, and in this PCR machine we need a DNA polymerase which is able to withstand a very high temperature. Once you understand the process, you will understand why. Okay, so now we are dealing with only the requirements. So understand, like Taq DNA polymerase, why it is used is an important question for your class 12 biology, because this Taq DNA polymerase is derived from Thermus aquaticus bacterium, which thrives under very high temperature, and its DNA polymerase is having the capability to overcome this high temperature or to work in this high temperature. If we just put our human DNA polymerase, it will get denatured. It cannot withstand the high temperature. So that is the next requirement. Okay. Then we have something called as dNTPs. dNTPs. You can see here, D I have written small letters, NTP capital letters. Okay. So here D stands for deoxy, N stands for nucleoside, T stands for tri, and P stands for phosphate. I have written here P's, phosphates, dNTPs, many dNTPs, that plural form I have written like that. Okay, so we will put it here as P, dNTP. Now what is this dNTP actually? If I am writing here ATP, this everyone knows. Can you tell me the full form? ATP? Yeah, it is adenosine triphosphate. Adenosine is a nucleoside. It is triphosphate, ATP. Now if I put here DATP, it will become deoxyadenosine triphosphate. That means this is part of DNA. If I put here small letter d, means it is part of DNA. If I don't put it, it is simply an ATP molecule. Okay. Similarly, I can have DGTP, deoxyguanosine triphosphate. Okay. Similarly, I can have DTTP and DCTP. But I will never take DUTP because we all know uracil is not part of DNA. So DATP, GTP, TTP and CTP. These are all the dNTPs. Okay, these are all the dNTPs. So that is also required. All these dNTPs are required for adding and forming a new DNA molecule. Okay, so Taq DNA polymerase is the enzyme which is needed for this addition, and the raw material here is dNTPs. Okay, now we were beginning with primers. Primer is the starting material required. It is a prime requirement. It is a starting material required. For example, we all know that our Taq DNA polymerase or our normal DNA polymerase can polymerize only in the 5' to 3' direction. It cannot do in the other way. That we all know. Okay, so here what happens is, these are the two strands. Okay, these are the two strands. These two strands get separated first, 5' to 3', and the other strand 3' to 5', and this primer gets added into the 3' region of each strand. 3' region. Okay, so what happens? The primer will begin with a 5' and end with a 3'. Here also it will begin with a 5' and it will end with a 3'. So now our dNTPs can be added like this in both the cases and it will extend. So primer is this one, which is added at the beginning towards the 3' end of the template strand. This is template strand. Towards the 3' end of the template strand, primer is added. So primer begins with a 5'. Okay, primer begins with a 5'. So usually you need two primers, primer 1 and this is primer 2. Two primers are needed for PCR. Okay, so PC primers will be very short oligoside bases are there which are added here. So these are the requirements: primers, dNTPs, Taq DNA polymerase, DNA template, and of course a PCR machine is needed. A small box like a PCR machine is needed as promised. In today's session, I'll take you towards the exact process what is happening in PCR. The whole process that is happening there can be just categorized under 3 steps. Okay, denaturation, annealing, and extension. Once we understand these 3 steps, it is very simple to understand the whole process of PCR. So let's look at the process. As I had mentioned in the previous previous session, I had taken you towards the different requirements for PCR. There I had mentioned like this PCR, the base is like our DNA replication. So once again, I would like to just bring it to you that DNA replication is, the video is already uploaded. You can just go through that, understand the concept, and then watch this video if you don't get clarity. Okay, so process here, like our DNA is having 2 strands, one is running from 5' to 3', another one runs from 3 to 5. Okay, so this is, I have just represented the double helical structure of DNA here. The first step in DNA replication is breaking off this hydrogen bonds and separation of the strands. Okay, there the enzyme helicase comes and does this. It unwinds the helix, the enzyme helicase. Here, there is no enzyme in PCR to unwind the helix. Instead, the helix are getting unwound by treating it under very high temperature. Under very high temperature, this DNA template strand is subjected to. So when it is subjected to very high temperature, what happens? The 2 strands gets separated. As a result, you can say it undergoes something called as denaturation. That is the first step. So denaturation is done using very high temperature. So once denaturation is done, how the strands will look like? The strands got separated. Now it looks like this. The 2 strands are now separated. Okay. Second step is annealing. Annealing means here comes the role of the primers. Regarding primers, I have just taken in the previous session what the primers are here in PCR when I took the requirements. Okay, if you have not watched, please go through that. So primers are added at the 3' end of the template strand. So this is our template strand. This is the 3' end of the template strand. 3' end here. So towards this region, the primer is added. And for this template strand, this is the 3' end. Here towards this region, the primer is added. So since this is ending with 3', usually the primer will begin with 5'. Here also primer begins with 5'. And we all know that DNA polymerase can polymerize only in the 5' to 3' direction. So for that, primer is added as a starting. Primer is added. So when the primer is added, that step is called as annealing. So instead of putting annealing here, for clarity, I'll draw once again and I'll just make it clear for you. Okay, so this is after denaturation, it looks like this. After annealing, still the strands are separated. One is running in 5' to 3', another one runs from 3 to 5. The strands are separated. But after annealing, what is happening is primers are added at the 3' end. Why I am drawing like this is because in your board exam, PCR is a very important question, and usually it will be asked like explain with the help of a diagram the 3 steps in PCR. So you have to be very clear. You have to draw the template strand first. Your first step is denaturation, after which how the strand looks like that you have to represent it. Followed by your second step is annealing. After that, how the strands look like, that you have to represent. Okay, and the third process is extension. Extension is using dNTPs and Taq DNA polymerase. You are extending the DNA. So how that is done? So here, before understanding what is dNTP and before understanding extension, you should know what is dNTPs and why we are using Taq DNA polymerase. For understanding that, please watch the previous video which I have put regarding the requirements in PCR, where you will come to know an important feature of Taq DNA polymerase. That is a polymerase which can withstand very high temperature. And why we are using it? Because in this first step itself, denaturation is done at very high temperature, and all this process are done in a PCR machine. Okay, so where the temperature will be very high, all these primers, this dNTPs, Taq DNA polymerase, DNA template, everything is put together inside that machine. So whatever we are putting, it should withstand the very high temperature. So that's why we have taken not the normal DNA polymerase but Taq DNA polymerase, which can withstand very high temperature. Okay, let's see extension also. So extension, I'm putting an arrow here. Okay, and this is extension. After extension, what happens? I'll just draw it here for you. So 3 steps. This is for easy for you to understand. I have just noted it down here. Okay, so after extension, how it looks like. So the same arrow I am putting here, this one. Extension here, using Taq DNA polymerase, the extension is done. Okay, first strand running from 5 to 3, second strand running from 3 to 5. Both the strands got separated in denaturation, and both the strands at the 3' end, they are annealed with a primer. Okay, now the dNTPs and Taq DNA polymerase have their roles to play. They come and extend here. So how do they extend? Suppose here it is adenine, thymine, guanine, cytosine, again thymine, adenine, adenine, like this. If it is there, the dNTP which is complementary to adenine will come here. So what will come here? Thymine. Again here adenine. So likewise the dNTPs will be added and till it reaches the 3' end. Here also, according to whatever is here, okay, whatever nucleotides are here, just complementary to that, the dNTP will come here and it will be added and extended till it reaches the 3' end. So now what did you get is actually you got here, this is one DNA strand, and you got here another DNA strand. We started with just one DNA strand here. This was our template strand. Now it got multiplied into 2 DNA strands. Okay, again this process will continue inside the PCR machine. So now this is already multiplied and it has become 2 DNA strands here. Now in the second cycle, what will happen is each of these 2, that means the first one, it will undergo denaturation, it will undergo annealing, it will undergo extension. So this one will again produce 2 DNA strands. Again this will undergo again denaturation, annealing and extension, and it will also produce 2 DNA molecules. Then each one of these will produce 2 DNA molecules. Likewise, the amplification process keeps on going as you set the number of cycles. Okay, so suppose you have put all the requirements inside the PCR machine and you have set the number of cycles as 2. Suppose you have set the number of cycles. I am putting here n as the number of cycles. You have set the number of cycles as 2. Then how many DNA strands will be there? It is using the formula 2 raised to n, you can get it. So where n is the number of cycles. So 2 raised to 2. After 2 cycles you will get 4 DNA molecules. Similarly 2 raised to, suppose after 4 cycles, that is 2 into 2 into 2 into 2. 2 into 2, 4 into 2, 8 into 2, 16. 16 DNA molecules you will get after 4 cycles. So after 10 cycles, how many you will get? You will get millions and billions. Okay, so likewise, how many DNA copies you want, that much cycle you will set it in the PCR machine, you will put all the requirements and things will easily happen inside that. So what is happening inside that we have learned now. So we have actually completed a process which will amplify the gene of interest. This word, please keep it in mind. This is an amplification method, and this is happening in vitro, means under controlled lab conditions, without the use of any organism, without the use of any bacterial cell or any vector or any plasmid. So we have learned today about an amplification method, that is PCR. I hope you all understood it, and if you have any queries, any doubts, feel free to ask. Thank you all.